Good afternoon, everybody. Thank you for joining us this journal club on month of April. Today, again, I am your only moderator, and you are safe from too much complicated papers. In the spirit of letting the stagiaire currently at Lyric is Jude has kindly accepted to present this paper from the UK about the changes in remodeling of the Purkinje network during heart failure in a model in rabbit. So I will I hope that Fanny will join us later, but now I leave the floor to Jude. Thank you.
Thank you. All right, so hi everyone. For those who don't know me, my name is Jude, and I'm currently doing my internship for my graduation of my Master 2 in cellular biology, physiology, and pathophysiology at the University of Bordeaux in the Lyric. And the main topic of my internship is around the Purkinje fiber and focused on trying to characterize the internal structure of the Purkinje fiber in young and old models related to its link with sudden death and the increase of arrhythmia in aging heart. And that's why I will present to you today an article around the Purkinje fiber and more precisely about the remodeling of the Purkinje fiber network in congestive heart failure in rabbits. So this article was published in 2021 in the AHA journals, which is the American Heart Association journal, which is a very good journal, as you can see, the impact factor of 9.7 this year and 10 for the past years. And that could explain the fact that the article is in fact very descriptive and don't really explain deeply the different parameters they are investigated. And that's understandable because there is a lot of rules when you publish in such journals. And that could explain the fact that there is a lot of references and the fact that the data supplement is very big and explain a lot of things that are not explained in the article itself. So this article is about the Purkinje network and the congestive heart failure. So I will introduce these two subjects before engaging the article. So the congestive heart failure can be defined by this citation, a complex clinical syndrome that results from any structural or functional impairment of ventricular filling or ejection of blood. This pathology has a high morbidity and mortality rate worldwide, where 50% of people will survive for 5 years and only 35 people will survive for 10 years. This is a progressive disease and death occurs through arrhythmias and pump failure. And the main characteristic of this pathology is inefficient myocardial performance, which means compromised blood supply. And that's why this congestive heart failure is classified with the left ventricle ejection fraction that leads to hypertrophy, like thickening of the ventricular chamber, and smaller filling capacity and ejection of blood. For the His-Purkinje network, this is a conductive network that allows the synchronization of the contraction by facilitating rapid and synchronous ventricular activation. It's divided that way. The sinoatrial node will initiate all electrical signals and transmit them to the atrioventricular node and transmit them to the His bundle. The His bundle will split in two branches, right and left, and these branches will spread inside and through the ventricle chamber, forming that way the left posterior bundle and the Purkinje network. And in the Purkinje network, we find the Purkinje fiber, which is a longitudinal assembly of Purkinje cells wrapped in sleeves of insulating of connective tissue. And you can see here an image I've got last year from a chip model where you can see Purkinje cells in purple, collagen and fibrocytes in green, and adipocytes in white. This cell, this Purkinje fiber is very specialized on conduction with specialized membrane electrical properties like fast action potential and high action potential amplitude compared with the malachite due to the electrical coupling allowed by connexin 40 and connexin 43. So for this article, they use one model and several techniques. The models they use are 44 male New Zealand white rabbits with 23 tests and 21 controls, and they induce the congestive heart failure by severe incompetent aortic valve by introducing a catheter via the right carotid artery. And after 3 weeks, an abdominal aortic constriction at the level of the renal artery, and that will induce the cardiac remodeling. They performed structural analysis with echo and ECG before and after the surgery, and also micro-CT or high-resolution X-ray computed tomography. But also metabolic analysis, that's not really metabolic analysis because they don't investigate the real function, but let's call it metabolic analysis, and with qPCR and IHC immunohistochemistry, and electrophysiology with sharp microelectrodes in tissue preparation. All right, so the first question is, does the model match the pathology, the congestive heart failure? And that's what they're looking at first. So I remind you the characteristic of the congestive heart failure, which are hypertrophy and structural or functional impairment of ventricular filling or ejection of blood. So we see here the control heart and the failing heart. We can see obviously that the failing heart are enlarged. And here we have the heart body weight ratio and the lung body weight ratio, which are all increased in the failing group. So we have our structural impairment. So we have through hypertrophy. And for the functional impairment, we have the aortic regurgitation, the mitral regurgitation, and the pericardial effusion, in addition with the left ventricle dilatation. And we have here the functional impairment, and they even get further with the impairment by looking at the ventricular internal diameters in systole and diastole, and some ECG parameters, which are all increased in the failing group, allowing us to say that the pathology is validated in the model. So after proving that the model is correct, we can analyze the structure of the Purkinje network to scout for remodeling proof. So first they look for the Purkinje network in the left ventricle and in the right ventricle, the septum and the volume chamber, the ventricle chamber volume, sorry. And we can see that the heart failure only affects the left ventricle, and the right ventricle remains unaffected. So they will look for remodeling in the left ventricle and in the left ventricle Purkinje network only since the right ventricle seems to be unaffected. So they look for the volume and the length of the Purkinje network. And long story short, they show that the Purkinje network in the heart failure is hypertrophied. The network is increased in volume, in free running length, and has longest free running segment length, allowing us to say that the heart failure impacts the Purkinje factor of the left ventricle, only of the left ventricle, through hypertrophy of the Purkinje network in volume and length. So we have our model, which is good for representing the pathology. We have they show that the pathology in fact affects the Purkinje through his volume, hypertrophy, and length. And now, what about the metabolic remodeling? And they investigate 40 transcripts. And this is an example of a table we can find in the data supplement. Of course, I will not get through all of these 40 transcripts. but I will sum it up as I can. So they analyze the remodeling of inward current carrying ion channels, potassic channels, calcium and sodium handling proteins, connexin, and pro-inflammatory and fibrosis. For the inward current of potassic channels and potassic channels, we can sum this up that these important channels for the action potential are all downregulated in the left Purkinje fiber compared with the control, so in the heart failure group. And it's the same for the calcic and sodic handling proteins and connexin. They are all downregulated in the left Purkinje fiber. We can highlight the fact that both connexin 40 and 43 are downregulated because they will push the analysis even further. First by the hypertrophic remodeling of myocytes, and then with the connexin protein expression. So with immunohistochemistry, they show that the myocyte diameter is increased in the left ventricle Purkinje fiber, in the left ventricle, but also in the right ventricle. We could discuss the fact that there is a big variability in the measures, and that the immunohistochemistry is not really optimal to quantify, but I will discuss this at the end of the presentation. We can say though that the orientation of the myocytes in the Purkinje network and in the ventricular wall is primarily responsible for changes in Purkinje and wall thickness. And then for the connexin protein expression, I remind you that the mRNA transcripts of connexin 40 and 43 were downregulated. We only see that the protein expression of connexin 43 is decreased in the left Purkinje fiber. That gives me the opportunity to remind everyone that the fact that a gene is downregulated doesn't mean that the protein expression will be lower. And that's why they wanted to see if the action potential of the Purkinje fiber was affected due to all the remodeling we saw in all the channels responsible for it. So they look for several electrophysiological parameters. First, they showed just here that there is no differences between the failing group and the control in regard of the ventricular rate. And then they look at, they investigate the action potential. We can see that the shape is very different. The resting membrane potential is more negative in the left Purkinje fiber, but high in the left ventricle itself, and the amplitude of the signal is higher in the left ventricle. So we have a difference between the left Purkinje fiber and the left ventricle itself, and they will investigate that later, but now on they look for the APD, the action potential duration, at 10, 50, and 90% of repolarization. The APD at 10% of repolarization is not affected by the heart failure, but the APD 50 and 90 are. These parameters are related to functional channels, such as APD 50 is related to phase 2 repolarization and so to calcic current, and APD 90 are related to phase 3 repolarization, such as potassic channel current. We can discuss the fact that they didn't really test the functional of it, like blocking a specific channel to see the very channel involved in this remodeling, but we still can say that the heart failure affects the calcic current and the potassic current. And as an end, they investigate the two APDs that were remodeled, APD 50 and 90, and the shape of the action potential in the transition from the left ventricle to the left Purkinje fiber. Of course, passing through the left ventral branches and the left Purkinje fiber left ventricle junction. So we can see that there is no differences between the left bundle branches and there's no differences at the level of the left bundle branches and at the level of the left ventricle in APD50 and in APD90 between the control group and the failing group. The only differences we can see is at the very level of the left Purkinje fiber and at the left Purkinje fiber left ventricle junction. We can see the shape shifting of the action potential here with only a landmark for be able to see the change. And we can see that the action direction is getting longer by the increase of the repolarization phases in accord with the increase of the APD50 and APD90. All right, so to conclude this article, in heart failure, there is a widespread remodeling of the Purkinje network at the structural level, molecular level, and electrical level. And this can lead to dysfunction and arrhythmias. They talk in this article about treatments that will forestall left ventricle dilatation, may prevent Purkinje remodeling, and are likely to benefit heart failure patients because they think that it could be the very stretch of the Purkinje fiber that due to the left ventricle dilatation that leads to the remodeling. Also, they pinned out an arrhythmia hypothesis, which they call the gay hypothesis. I'm not a specialist in electrophysiology, but they talk about it, that they explain that the reentry and the arrhythmia could be caused by this gay hypothesis. In fact, they say that the conduction of the action potential could be only in one branches and not on the other one. And when the first potential action potential activates the ventricle, it could retrogradely propagate through the other branch and create a reentry phenomenon. But what I think is the most impressive in this article is the relation they succeed to show between the Purkinje fiber and pathology such as heart failure, because it is very difficult to analyze the Purkinje network. There is a lot of limitations such as the tissue availability. It only represents 2% of the heart mass with complex various heterogeneous structure. The electrophysiology analysis are complex to perform due to the specificity of the conduction in the fiber, and the markers are limited, species dependent, and not variable in humans. And moreover, the variability in the incidence, the size, the shape, and the composition of the Purkinje fiber is very high and could explain the variability in the measures. Thank you for your attention.
Thank you, Jude. Very nice presentation, concise and clear on the paper.
I hope that was clear.
Yeah, it was very clear. Thanks. I don't know why I see my face instead of your screen. Just one small remark on my side. When you mentioned that the 40 biomarkers were on the metabolism panel, there's very little metabolism there. It's mostly a lot of ion channels.
Yeah.
And, maybe this is the only thing that I could see even when I was reading the paper is like the sodium hydrogen exchanger, but that's like okay, that's just basic pH. It can mean for anything. Very interesting, the story of the hypothesis of the gate. Don't know if you remember from your theoretical studies. This is the issue that can lead in this case, as you can see, there's a what is called an AVNRT, so atrioventricular node reentrant tachycardia.
Yes.
or you can have AVT, so you can have an accessory pathway which does not pass the AV node, but re-enters in the atrium. And yeah, definitely quite interesting. Jason has a question. Please go ahead.
Yes, excellent choice of paper. I really enjoyed that. So one question for me is, how deep in the endocardium do Purkinje fibers penetrate in the rabbit heart? And then this will lead to my second question.
Sorry, I don't remember it's sub just under the skin, not very profound.
Because how far they go in is interesting when you're comparing the links of APD and the endocardial LV versus Purkinje, because the depth, there's been a lot of research, I don't know in rabbits, but in humans, they show that during heart failure, only APD in the epicardium gets longer, right?
Yeah.
So if the PMJs penetrate closer to the mid of the thing, you're not really comparing.
So, yeah.
You won't see a change on the epicardium of APD, and then you're making all this analysis, but you will the further that you get in, and the influence and the interplay between the network and the Purkinje fibers might be dependent on the depth. That's just more of a comment on part of the variability that they may see, because the variability may be dependent on that. And just to be aware that in humans, APD is different across the heart, and the gradient changes differently within heart failure. Only prolongation in the epicardium. And my second question, I guess actually a third, sorry, is when you looked at the Purkinje volume, what's the explanation for increased volume? I can understand the length changes based on the network maybe getting stretched due to hypertrophy, whatnot. What is the explanation for the increase in the volume?
In fact, when they doing this, it's on the data supplement, they also did electronic microscopy. And so they are taking the 3D of the fiber and analyze it by the short axis of the biggest fiber they found. There is an image in the data supplement, but I didn't put it. But it's mostly the electronic microscopy that allows them to say that there is an increase in the volume, and not really the microscopy.
Because I mean, it might be they're just pulling more tissue into whatever the area they're measuring. You know, the network's changing and things. I'm not so sure.
Yes, but I don't even know how they managed to like get the same shape of network. I mean, the variability is very high, so
yeah. I agree with you. There's a little skepticism there for me. Thank you very much. Thank you.
No problem.
Yes, we have another question.
Okay.
Chantal, go ahead.
Okay.
Hi, my name is Chantal Conrad, and I'm a cardiac electrophysiologist from Amsterdam. I'm working together with Ruben. So thank you for having me also in this journal club. Very nice presentation and very structured analysis of the article, which was a massive one. I have one question. In the supplementary paper, there was a figure 5 in which they were looking at the Purkinje cells and noticed that there was cell damage, also a lack of integrity of the nuclear membrane of Purkinje cells. And I was wondering, is it somehow, could it be that it's still some kind of acute injury effect on the Purkinje network that we're looking at, and not so much a sign of chronic heart failure, but more a sign of acute injury on the endocardial part of the left ventricle?
Yeah, it's absolutely possible. Yeah, because the stretch they are pulling for the Purkinje network is very strong. So it could be. I think they discussed this in this article. When they said this, they said that all the results and the remodeling could be linked to the stretch of the Purkinje during the left ventricle dilatation, but not really from the heart failure itself.
Yeah, Also because in the left ventricle muscle, they also didn't find a lot of remodeling things like for instance in fibrosis or other remodeling markers were not upregulated, if I remember well. So maybe it is still, even though it's something like 5 weeks or so after start of the aortic regurgitation and later the binding of the aorta descendants, maybe it's still more a model of acute heart failure than of chronic heart failure, I was wondering.
Yes, maybe. It could be an acute heart failure, yeah. But I guess it's, for me, in my point of view, it's still that like the model is representative of the pathology. Like I'm not really sure if we can say that, but with the test they did, I think it's representative.
I surely agree that it is representative of heart failure. I was just wondering which stage of heart failure it is representative of, because of the conclusions they draw also for instance proximal pacing in the His bundle area or so, that would not be effective. I think that maybe you cannot really conclude this on basis of this model, but it's absolutely very interesting, the findings. Thank you.
Yeah, I totally agree. Yeah.
Okay. Cool.
Yeah, it's interesting, this figure 5, as you were mentioning, especially because I learned the new things. I learned that there can be intranuclear mitochondria. It's really nice, and apparently it's a marker of congestive heart failure in around 6% of patients. So very, very interesting. I wonder what are they doing there? How did they get there? And what are they doing there? Really interesting. This model, in my opinion, is it's very acute. So we might look at a very early stage of
because three weeks is short.
Yeah.
All right.
Sure, they exaggerated, so the phenotype might be severe, but still in the early stages. The electron microscopy images are interesting, as usual, fairly limited because it's a work per se to analyze only electron microscopy. Very interesting in the I think that from the micro CT point of view, they consider the whole volume of the free running fibers, and they concluded that the length and the volume was increased significantly in this model of heart failure. It's pretty straightforward. Why they are growing? Can you show me again, please, the figure on the immunohistochemistry when they show the size of the cardiomyocytes? The one.
Oh yeah.
Yeah.
That one.
Yeah. So let's debate on whether immunohistochemistry done like this it's a relevant way to assess cardiomyocyte diameter, especially in such an heterogeneous tissue. I am very skeptical. I used it myself, but not in these conditions, and only to measure cardiomyocytes in general, and you have a lot of chance of running into perpendicular bundles, and then you can measure quite nicely the diameter.
Okay.
Does this make sense to you or to the audience to do it in immunohistochemistry? For example, like in the RV section, the lower left figure, I don't think they are perpendicular. Not all of them, at least. So can be.
Yeah, they talk about that in the description or in the data supplement. I don't remember, but they think that they took around 10 sections of our test section across the cross section, and they assume that it's a transversal cut, by taking always the minimum diameter to compensate a potential oblique sectioning. So we're not really sure of that.
It leaves a lot to interpretation, in my opinion. But okay, it's one way to do it automatically, and then you trust the process. Well, I would prefer, in general, I would prefer to use something that is not bound to glass, or something with a little bit of thicker section and confocal. I don't know what is the audience opinion on that. A very interesting question from Jason, like how penetrating is the Purkinje network in these animals? This is really, I don't know. In the sheep and in the pig, we know that it's like everywhere. In the human is mostly endocardial. There's a very nice picture. Maybe we can comment on this. I will just share my screen, or if you want to, do you have the supplements, Jude?
I think, yeah. But I would just to talk about one thing about my project, because they use the neurofilaments to highlight specifically Purkinje fiber. For my project, it could be a great way to visualize them in immunohistochemistry. And I don't know if you know if this is a good idea or.
Never seen it. But if you are thinking about using Connexin 40 or Connexin 43.
Yeah, we already used that, but it didn't work.
Yeah, that's why apparently nothing works in sheep, and it can be your next best guess as a marker. I have to say that these pictures could be better done. You can see the orange as a superposition marker, but a panel with single colors would have been more clear to us to understand what is the level of colocalization, what is the level of, but I mean, you can say the orange are your Purkinje fibers, or are your Purkinje cardiomyocytes.
Okay.
Guido and Jude, do you hear me?
Yes,
yeah.
I'm Melissa speaking, sorry. Do you think that in that case, as you nicely said earlier, that electronic microscopy could help to better really quantify the myocyte diameter as well as Purkinje fibers, or?
Do I think it's relevant? I don't really understand the question, sorry.
No, it's Menez was inquiring if the electron microscopy adds significantly to the characterization of the diameter or the structure of the Purkinje network.
And even of course in the myocyte diameter, but also how actually the Purkinje is remodeled during hypertrophic cardiomyopathy, because we can see that in hypertrophic cardiomyopathy, we do have patients that present immediately with ventricular fibrillation and not ventricular tachycardia that degenerate into VF. So it indicates that maybe, as you say, the diameter of this Purkinje increase, but also what is its interaction with the surrounding myocyte.
Yeah. From what I can see from their supplements, the electron microscopy is fairly limited in what it can do. Of course, it can confirm the diameter because it allows you to do 3D reconstructions, and it gives you like a very nice insight on the
I have it.
Possible functioning. Yeah, if you can take this figure 5, figure which one is it on the?
Yeah, of course, it's always a link between
Figure 5.
structure
Yeah.
This one?
It is increase, modification of the orientation of fibers, whatever, and then functional to see what happens, of course, in this Purkinje and especially at the Purkinje myocyte junction.
I think what is very, very interesting, if you want to check the changes in volume, confocal microscopy might just be enough, especially if this neurofilament marker works. If nothing works, then you are bound to electron microscopy. That's right. As Jude was mentioning, the antibodies, no antibodies for connexin 40 seem to be working. This is something I discussed also last week with Marion about this. And so if this neurofilament marker works in combination with the NCX1, then confocal might be enough. And electron microscopy in this case will give you just more information on the sarcomere, glycogen accumulation, and mitochondrial network. What is very interesting in this paper is this study on the this optical mapping of the running fiber that shows the junction, the left ventricle, and the free running fiber. Jude, could you please show again this slide? I think
So,
was
sorry, I arrived late. Sorry for that, but yeah, I would like to see this.
Yeah.
Which one?
The last one where they showed the optical map of the that one. Yes, the optical mapping of the running fiber and the junction. This is really, really interesting, really elegant. And in their supplements, it's even more elegant. They did a better job in the supplements. It's much more clear. There is an APD. There is more clear what is happening at the left bundle branch, at the free running fiber, and the junction in the left ventricle. Very interesting because the left ventricle seems to be not affected in terms of APD, but the conduction system and the junction are highly remodeled.
Yep.
Okay. Is there any other question for this paper? No. Okay.
Can I ask one?
Please, Andre, go ahead.
Yeah, sorry, maybe it's irrelevant, I don't know, but I'm curious. Because as far as I understand, there is two types, right, at least, of heart failure. It could be with reduced and preserved ejection fraction. And I'm not sure, do they say something about this? And because as far as I understand, also there is a bit different strategy of treatment and diagnostics and so on and so on. So, and probably some difference in Purkinje network changes. So do they say something about this? What kind of heart failure did they made in the end?
You talk about the treatment they foresee?
Sorry?
Sorry, I think they don't talk about other type of heart failure and the congestive heart failure. I'm not sure if.
So what was? Can you just summarize for me what was the model of heart failure?
It was rabbit. Was all right. I could put you again.
Sorry again. I just missed your talk. I'm sorry.
No problem. The rabbit. 21 congestive heart failure.
Aortic valve. So this is Andre. To answer to your question, it's more likely reduced ejection fraction.
It is.
Mm-hmm.
It is.
Okay.
Later, they show the fractional shortening in the ejection fraction, which is reduced.
Yeah.
About 10,
because they called this just heart failure and didn't specify. So I'm curious.
Based on the model, it's reduced.
And remember that models of preserved ejection fraction or moderated reduction, they are very few. Very few.
Which I found strange. Jude, can you show us again the optical mapping with the action potential in control and in heart failure? What I found strange is even in control, more or
This one?
less the APD,
Yeah.
the APD, it looks like you see you start at 250 from the branch, right? And it is reduced until 200 until the junction. I mean, we know that the Purkinje, especially at the junction, has the longest APD, just to prevent any re-entry. And in that case, in blue, when they are at the junction, you see that in red it's increased, definitely.
Mhm.
But in control, I mean, there is not a big difference from the upper tracing and the lower tracing, isn't it? Or
You talk about the slight decrease.
if you look just below the morphology or action potential,
Mhm.
that comes from the proximal part towards the junction, the what it is, here.
Yeah, the free running fiber. Yeah.
Yeah, normally you should have here a very long APD, right?
Yeah, the proximate fiber action potential is very much longer than the ventricle, but maybe it's due to the models they use.
No, but I mean it's control. So control, even
Yeah.
though the APD is very short because it's a rabbit model, but still, I'm very surprised.
I have an extra. I don't know if I put it up, and sorry, I thought I have an extra images for this, but I don't.
Okay.
It might be too small for us to appreciate by eye, and the model might be this might just be a representative figure which doesn't show it properly.
But I agree, there's, in 1970, what the studies by Meyerberg show that the APD at the junctions near the PMJ is the longest.
Yes.
And now, what confirms this clinically? So because those are probably isolated things, so maybe the influence of the myocardium in the rabbit or in ARP, I don't know what they're doing, has an influence on the Purkinje network through retrograde propagation. But
Yeah, sure.
yeah, but I'm just curious, has this been verified clinically as well, that this would be the case?
No, Jason, no.
Yeah, because I mean,
because we lack technique to map the.
Yeah.
I have ideas, but we lack technique to really. Yeah, we were discussing a lot with Ed on that topic, and I have a project. But maybe we can discuss about this with you too. But yeah, the problem is how you identify in clinics this kind of characteristics. It's very difficult.
Yeah, because when everything's intact versus isolated, I have no idea.
Yeah.
All right. We didn't stay a lot on this. I invite you to look at the paper where they discuss about these mRNAs expression modification. And you will see better why the APD50 and the APD90 are mostly deregulated. Also, the panel of potassium channels is quite, and subunits is quite exhaustive. So you will easily be convinced that it is mostly. It's also calcium related at the level of 1.2, 1.3 as well. That is interesting. Only on the Purkinje network, but it's mostly potassium related. But you will see it. Right. So we are very nice in the time limits. So if there is no other question, I would like to thank you all. Thank you, Jude, for this excellent presentation and discussion.
Jude,
so much.
can you send me your presentation, please? I can.
Of course.
It. Thank you so much. I think I just send you something in the chat. You have my email address.
All right.
And Jason, can you just stay connected while maybe everybody is disconnecting? Jason, you have left already?
No,
Oh,
I have not.
Okay, right.
I'm still here.
Okay, and I think there is Laure still. I need to talk to her. Thank you.
Okay, thank you very much, everybody. We will see each other next month.